All right, hello everybody. I'm going to go through transcription and translation with you and make sure you know everything you need to know about transcription and translation so you can do your project and you can do well on the quiz next Friday. So here we have transcription of DNA into RNA. So transcription is using DNA as a template to make your RNA. Remember, DNA has T's, RNAs have U's. So you got to be careful when you're matching up the bases that you do it correctly. So now, this is RNA polymerase. You don't need to know that for the sake of this class, but you need to know that a protein, this is a protein, comes in and helps with this process. Now this protein, super tiny, is within the nucleus of the cell. So the nucleus within a large cell. So cell, nucleus, protein, DNA, transcribing into RNA. So for here, this protein will start to open this up, separating the DNA strands. This is important because that's how we actually match the bases together. So now this enzyme also puts the RNA nucleotides. Remember, these are nucleotides. Those are the building blocks of DNA, and they're different colors for the 4 different bases that we have in DNA. So here, it opens it up, and then we match it one by one to the RNA nucleotides. So here, T is going to pair with A in RNA. So I'm going to attach it there. A is going to pair with U in the RNA. Make sure not to do the T because that's only DNA, and we're making RNA. The C pairs with the G. All right, keeps going through. And notice at this point, the DNA is actually going to separate immediately from the RNA. So the DNA is going to keep going. The RNA is going to split off and become its own thing. Then we have G pairing with C. Remember, this is just like we were doing on our paper in class yesterday. T pairs with A. Keep going. Remember, A pairs with U because this is DNA to RNA. And it goes through one at a time and keeps adding them. A with U. Keeps going. C with G. C with G. Keeps going. Notice on this side, the DNA is starting to come back together. That's exactly what happens. DNA only zips open briefly to allow the RNA nucleotides to match with the DNA strand. So next, A pairs with U. All right, DNA is starting to zip back up. Gonna bond between strands. C pairs with G. A pairs with U. This still is coming together as it's going along. Okay, G with C. Sorry, this has gotten a little off center. G with C. T with A. Look, we're almost to the end. And then they combine. It's amazing. Every single cell, not only in humans, but every single living thing does this process. So every single living thing needs proteins. Right, we keep going. And that's zipping back up. T pairs with A. And G with C. And then this just comes through. DNA zips back up. Bonds together. And voila. You got your DNA strand, and this will leave that protein. And now it is free floating in the nucleus. Now the mRNA needs to leave the nucleus in order to go to our next step, which is called translation, and I'll make a new video for that one.